classified as living things since they lack many characteristics of life, but there is some debate in calling them non living as well. Medications like antibiotics attack bacteria like E. coli, which are living cells. Antifungal medications can be used on fungi like athlete's foot, which are made of living cells. Viruses are not bacteria or fungi. You can't classify a virus as a prokaryote or a eukaryote either because it's not a cell. So, what is their structure like? First of all, viruses are much, much smaller than cells. You typically are going to need an electron microscope in order to see a virus. Viruses come in many shapes. Our favorite one to draw is the bacteriophage. It sounds very fancy, but it's a type of virus that tends to attack bacteria. Despite this being our favorite to draw, know that the shape of viruses vary greatly. One thing all viruses have in common is that they have some type of genetic material. This genetic material can be in the form of DNA or RNA. Viruses usually have some kind of protein coat, also known as a capsid. The capsid can protect the DNA or RNA genetic material. Viruses can also have other structures. Some viruses package special enzymes with them. Some viruses have an outer envelope. These additions can be useful to the virus in virus replication. One thing that makes virus replication or reproduction unique is that they can't replicate without a host. And it's their reproducing that tends to be a problem for its host, whether the host is you, in the case of the common cold, or an insect, plant, this bacterium. Viruses tend to be very specific for the host that they're going to infect, but anything that is alive can be a host because cells have the machinery that viruses need in order to replicate. We're going to talk about two types of viral replication cycles. The first one is the lytic cycle. In the lytic cycle, the virus, remember, very selective, attaches to a host cell. It often binds to a receptor that this cell has, which gives it access to attach. The virus can inject either its DNA or RNA, depending on what kind of genetic material it has, into the cell. Some types of viruses are actually taken inside the cell themselves. Now, you would think that the cell would notice viral DNA or RNA or an entire virus that has been taken in, but in many cases, it does not. It takes the genetic material from the virus and it starts following the instructions, which in this case is very bad because the instructions tell it to make copies of the virus. The cell uses its own resources to start building. It starts making so many copies of the virus that it can cause the cell membrane of the host to rupture, explode, lyse. So what happens is now that these new viral copies, they can get out of the cell and go and infect other cells. This is known as the lytic cycle. By the way, the lysing of the cell membrane is a very bad thing for the cell. The cell cannot survive without its cell membrane. The other cycle is called the lysogenic cycle, and I like to remember this is the longer word, so I like to think of